To heat our houses in the winter, we burn fuel. The fuel breaks down chemically and releases energy as heat, which heats up the air particles in the room. The process of burning is made up of chemical reactions, such as breaking the bonds within one molecule and making bonds with other molecules, which releases energy. If the energy required to break bonds is smaller than the energy released when new bonds are formed, then this net energy difference is released into the environment. This is called an exothermic reaction. All organisms on Earth use a fuel called ATP to power almost all of the reactions that take place inside them. These reactions are often essential and range from doing mechanical work like muscle contraction, to transporting molecules across membranes, to synthesizing new molecules. But what is ATP, and how does it store and release energy? ATP is a small molecule that's made up of three separate components. It has an adenosine, a ribose, and a tail that consists of three phosphate groups. Thus the name adenosine triphosphate. The breaking of the bonds between the phosphate groups and subsequent formation of new bonds with other molecules like water allows ATP to release energy via an exothermic reaction. Cells can then use that net amount of energy released for vital chemical reactions that keep us alive. The resulting products are adenosine diphosphate, or ADP, which has just two phosphates, and an inorganic phosphate group. So, ATP can be broken down into ADP and inorganic phosphate, but ADP can also combine with inorganic phosphate to reform ATP, making this a crucial reversible reaction that allows the molecule to be reused as a source of energy. How is this crucial molecule made? Inside most cells exist cellular compartments called mitochondria. They have an outer and an inner membrane, the latter of which surrounds the matrix. Within the inner membrane sit thousands of enzymes called ATP synthases, which synthesize the vast majority of the ATP molecules in our bodies. ATP synthase is made up of many different subunits. The enzyme can be split up into a head region, which sits within the mitochondrial matrix, and the membrane region, which is embedded in the inner membrane. In the head region of ATP synthase are 3 alpha and 3 beta subunits, arranged in an alternating circular manner. Each beta subunit cycles between 3 different states to make ATP: open, loose, and tight. If we focus on just one of the 3 beta subunits, we will see that ADP and inorganic phosphate are able to bind because they are complementary to the shape of the enzyme in the loose state. Then, something causes the loose state to suddenly transition into the tight state. During this transition, ATP is formed. Now, something else causes the tight state to transition into the open state, which releases ATP from the enzyme. All 3 beta subunits simultaneously undergo this mechanism, but are always in different states to each other. What causes the transition between the states of the beta subunits, allowing ATP to be synthesized from ADP and inorganic phosphate? The alpha and beta subunits surround a protein shaft called the gamma subunit, which rotates and causes the beta subunits to change states. More specifically, its asymmetry means that it interacts with each beta subunit differently depending on its orientation, which changes the state. One full rotation of the gamma subunit forms 3 ATP molecules. But why does the gamma subunit rotate in the first place? At the base of the gamma subunit is the C ring, which is embedded in the inner membrane. In humans, the C ring is made up of 8 C subunits which form a circle, and is attached to the A subunit. It has been discovered that rotation of the C ring also rotates the gamma subunit, as the two are linked. How does this happen? In every C subunit is a negative charge, which can gain a proton to become neutral. In this diagram, we can see that the A subunit has 2 channels, one facing the intermembrane space and the other facing the matrix. The intermembrane space has a high concentration of protons compared to the matrix, so one by one they enter the A subunit to protonate a negative charge. Neutral charges become negatively charged again when they lose a proton on the opposite side of the ring via the second channel. In the A subunit is a positive charge, which is attracted more to the next negative charge than the neutralized charge, like how opposite poles of a magnet are attracted. Attraction of the positive and negative charges pulls the C ring to turn it clockwise, and the whole cycle repeats when another proton is taken up. The fascinating mechanism of ATP synthase works through C ring and gamma subunit rotation, which drives the beta subunits to produce our vital ATP. Without this endlessly turning nanomotor, life as we know it would not exist.